So this is the third lecture on development of the brain spinal cord and pituitary gland. So here we'll be seeing the development of the brainstem and cerebellum. So rhombencephalon, otherwise called the hindbrain, it has two parts, myelencephalon and the metencephalon. Myelencephalon is the caudal part, forms medulla oblongata. Metencephalon is the cranial part, which forms pons and cerebellum. The fusion between mesencephalon and rhombencephalon is marked by constriction called rhombencephalic isthmus. The cavity of the myelencephalon forms a part of the fourth ventricle. You can see here. So this is the mesencephalon, this is the metencephalon, and this is the myelencephalon. Myelencephalon forms the medulla, whereas the metencephalon forms the pons and cerebellum. The cerebellum is being developed from the rhombic lip. You can see this is the rhombic lip. So this rhombic lip, you can see here, this forms the cerebellar plate. The central part of the cerebellar plate forms the vermis, whereas the peripheral parts will form the cerebellar hemispheres. You can see the developing cerebellum here from the rhombic lip. You can see the pons developing from the metencephalon. You can see the medulla developing from the myelencephalon. The sulcus limiting divides the mantle layer into dorsal alar lamina and ventral basal lamina. The roof plate of the cranial part of the medulla widens to form the roof of fourth ventricle. The basal lamina of the myelencephalon differentiates into the following columns: the general somatic efferent, which forms the hypoglossal nucleus supplying the muscles of the tongue. Special visceral efferent is the nucleus ambiguous for muscles of the third arch glossopharyngeal nerve, muscles of the fourth arch superior laryngeal nerve, and sixth arch recurrent laryngeal nerve of vagus via spinal accessory nerve. And special visceral efferent, dorsal nucleus of vagus and inferior salivary nucleus for parotid gland through glossopharyngeal nerve. And you can see the cells from each alar lamina migrate ventrally in the marginal layer to form the bulbopontine extension and finally develops into the olivary group of nuclei. As you know that inferior olivary nucleus, dorsal olivary nucleus, all those develop from this. So remaining cells of the alar lamina differentiate to form the following columns: general visceral efferent, dorsal nucleus of vagus, special visceral efferent. Sorry, this is actually afferent. It is not efferent, it is afferent. General visceral afferent, special visceral afferent, then general somatic afferent, and special somatic afferent. So you can see the general visceral afferent is the dorsal nucleus of vagus. Special visceral afferent is the nucleus tractus solitarius to receive taste sensation via the 9th and 10th cranial nerves. And general somatic afferent is the spinal nucleus of the trigeminal nerve, nucleus gracilis and nucleus cuneatus for tract of Gall and Bardak. And special somatic afferent is the cochlear and vestibular nuclei. The white matter of the medulla oblongata is mostly derived from ascending and descending tracts. So then formation of the tila choroidea and choroid plexus. The widening of the roof plate of myelencephalon results in formation of the single cell layered ependymal roof plate of fourth ventricle. This roof plate is surrounded by vascular pyomatter called tila choroidea. The vascular proliferation of pyomatter invaginates underlying ependyma. So thus formed vascular tuft covered with ependyma are called choroid plexus. Choroid plexus produce cerebrospinal fluid. Tila choroidea ruptures to form the foramen of Lashka that communicates the fourth ventricle with the subarachnoid spit and also the foramen of Magendie. So you can see here. So this is the fourth ventricle. This is the roof of the fourth ventricle, which gives rise to the tila choroidea and choroid plexus. So then development of pons. The metencephalon will form the pons and cerebellum. The metencephalon is divided into ventromedial part, which forms the pons, and dorsolateral part called rhombic lip, which develops into cerebellum. The pontine flexure separates pons from the medulla oblongata at pontomedullary junction. So bulging basilar part of the pons shows midline groove called basilar sulcus. The pontocerebellar fibers form midcerebellar peduncle. The dorsal sulcus shows elevation in the floor of the fourth ventricle as facial colliculus. You can see here. So this is the cerebellum, which is being developed from the rhombic lip, and you can see this is the pontine nuclei in the basilar part of the pons. So and you can see these are the different columns in the basal lamina and alar lamina. You can see here general somatic efferent column is the motor nucleus of the abducent nerve, and you can see the special visceral efferent column, motor nucleus of the trigeminal and facial nerve. General visceral efferent column, namely the superior salivatory nucleus and lacrimatic nucleus. Special visceral efferent, nucleus of tractus solitarius. General somatic efferent, main sensory nucleus of the trigeminal and nucleus of spinal tract of trigeminal nerve. And special somatic efferent, vestibular and cochlear nuclei. So formation of pontine nuclei. So as we have seen earlier, the formation of the inferior olivary or the olivary nuclei, same way from the bulbopontine extension, the cells will migrate cranially into basilar part of pons to form the pontine nuclei. So basal lamina will have these three parts, namely the general somatic efferent, motor nucleus of the absent, special visceral efferent, motor nucleus of the fifth, and facial nerve. In the general visceral efferent, suprasalivary nucleus and lacrimated nucleus. So the alar lamina will have the following things, namely the nucleus tractus solitarius, principal sensory nucleus, and nucleus of spinal tract of trigeminal nerve, vestibular and cochlear nuclei. The rhombic lip in the dorsolateral expansion of the alar lamina of metacephalon. So this rhombic lip forms the cerebellum. You can see here, this is the development of the midbrain. So this is the basal lamina, this is the alar lamina. So this basal lamina is actually the motor component. The alar lamina is the sensory component. You can see the alar lamina differentiating into formation of the colliculi, and even the alar lamina will also form the red nucleus and substantia nigra. This basal lamina will give rise to the oculomotor nerve nucleus, then the trochlear nerve nucleus, and the Edinger-Westphal nucleus. So midbrain develops from mesencephalon. The sulcus limitans divides the mantle layer into dorsal alar lamina and ventral basal lamina. Basal lamina, along with the floor of mesencephalon, forms tegmentum of the midbrain. So basal lamina of midbrain differentiates into the following columns: general somatic efferent column, oculomotor trochlear nucleus of the extraocular muscles, general visceral efferent Edinger-Westphal nucleus of the third nerve. Then alar lamina gives rise to stratified zones of colliculi, superior and inferior colliculi. So superior colliculi is concerned with the visual reflexes, inferior colliculi is concerned with the auditory reflexes. The cells of alar lamina migrate ventrally to form red nucleus and substantia nigra, as I've showed earlier in the diagram. Remaining cells of alar lamina along with the basal lamina form periaqueductal gray matter. So alar lamina, you can see the general somatic efferent, the mesencephalic nucleus of the trigeminal nerve, and special somatic efferent, the superior colliculi reflects center for light, and inferior colliculi reflects center for hearing. So white matter, the formation of the crus cerebrae, most of the descending fibers pass through the marginal zone of the ventral part of the midbrain that forms the crus cerebrae. The cavity of the mesencephalon is the cerebral aqueduct. So then development of cerebellum. So as I said earlier, the rhombic lips are the main things which forms the cerebellum. So bilateral rhombic lips arising from alar lamina of metencephalon form cerebellum. Rhombic lips produce bulging that fuse in the midline to form cerebellar plate. The cerebellar plate differentiates to form vermis and cerebellar hemisphere. Some cells of the mantle layer migrate outwards to form the external granular layer, whereas remaining cells of the mantle layer form the cerebellar nuclei, the dentate nucleus, emboliform lobus nuclei. External granular layer differentiates to form the cerebellar cortex. The axons of the dentate nucleus form the superior cerebellar peduncle. Axons of pontine nuclei form middle cerebellar peduncle, and axons of olivary nuclei form the inferior cerebellar peduncle. You can see here the metencephalon, proliferation of cells of rhombic lip, fusion of the rhombic lips in midline, formation of the cerebellar plate, primitive cerebellum, differentiation, the central vermis and bilateral cerebellar hemisphere, development of the fissures, separation of the lobes and lobules. In three layers of the rhombic lips, the cerebellar nuclei, dentate, emboliformis, globose, and fastigious, they develop from the mantle layer, and a few cells from mantle layer migrate towards superficial part, and they form the external granular layer, which gives rise to the cerebellar cortex, astrocytes, and oligodendrocytes. You can see this is the cerebellar plate, the vermis, and the cerebellar hemisphere. You can see the developing cerebellar hemispheres. You can see the flocculus. You can see the middle lobe. You can see the anterior lobe. So you can see the differentiation of. You can see here, this is the mantle layer. Few cells have migrated to the surface, forming the external granular layer. And you can see the external granular layer differentiating into the molecular layer, Purkinje cell layer, and granular layer. And you can see the remaining part of the mantle layer is forming the deep cerebellar nuclei, namely the dentate nucleus, emboliform nucleus, globose nucleus, and fastigious nucleus.